Well, thanks for joining me today for our training topic on fuel microbiology made simple. This is a streamlined version of a longer belt performance training that we called fuel microbiology, everything you ever needed to know but were afraid to ask. And that training clocked in at about 2.5 hours. But even then, that length didn't really do the topic justice because for all the details that we could cover in that long-form training, we were still, really only going to scratch the surface of what there is to know about fuel microbiology and how it affects us. So in our time together today, what we want to do in this more streamlined coverage of the issues is our goal is to come away with a clearer picture of the basics of fuel microbiology and fuel microbes. What they do in storage tanks, what they do in the water that's found in storage tanks, the effects that they have on the environment around them, and how they cause problems. Then we'll transition into considering the monitoring and sampling issues or considerations that we need to take into account as we seek to stay on top of whatever problems we have identified. Then lastly, we will address the what we need to do about it issues, summarized as contamination control, which incidentally involves more than just dumping biocide into a tank. It's a little more complicated than simply something like that. That is, if you want to do it well and do it effectively. So let's kick things off by talking about the basics of fuel microbiology. And to do this, we start by considering why this is or should be important for stored fuel users, because it is a costly problem. The costs of this issue are pretty substantial. A 2002 corrosion study from the Federal Highway Administration cited that the nationwide cost of corrosion damage was more than $270 billion per year. On the field level, like at a gasoline station and the C-store, you have financial consequences like losing between 5 and 20% of their fueling revenue due to slow flow rates caused by corrosion. Not to mention instances of catastrophic repair costs starting at $200,000 to $500,000 for a storage tank leak due to corrosion damage. So left uncontrolled, microbial contamination in fuel systems is linked to a number of expensive problems. So it is essential that we have a basic understanding of the key tenets of the issue. So to kick it off, we are going to talk about fuel microbiology concepts. Now, starting very simply, we all should at least know that microbes are single-celled organisms. They're ones like bacteria and viruses and fungi and archaea and algae and protozoa. Those are all microbes. And you won't find all of these kinds in every single place. Like it's rare to find, say, protozoa or archaea in fuel or fuel system samples. So when we talk about fuel microbiology, we concentrate on the biggest problem solvers there, which are bacteria and fungus. Now, even among these basic categories of microbes, there are subcategories. And subcategories tend to reflect where you're likely to find them in a fuel system and the kinds of things that they need to survive and the kinds of things they do. So you've got aerobic versus anaerobic, which relates to how much oxygen they need to survive. You've got microaerophiles, which are ones that need a little, some oxygen, but they don't need an oxygen-rich environment, and they can't survive in an oxygen-free one. There are facultative anaerobes, which are ones that can kind of switch between one mode to the other. They start off thriving in oxygen-rich conditions, but then they deplete the oxygen and turn it into an oxygen-free environment, which then allows other kinds of microbes to come in and grow. So they kind of play a helpful process there. So microbes are very good at adapting to the available conditions. In fact, we can classify them further based on how they use their environment, whether they get their energy from light or from breaking down chemical compounds. In order to get the carbon that they need, do they break down organic molecules or do they use gases like carbon dioxide? Now, in a given fuel tank, and this is important to understand, in a given fuel tank, you can have thousands of different kinds of microbes, and they're all living symbiotically. So they kind of require the presence of each other in order to grow and best thrive. So that is why microbiologists will talk about it like a microbial colony. They'll talk about it like it's its own little ecosystem, so to speak. And we're just now coming to more of a realization that things are a lot more complex than we first imagined. But that pales in comparison to the simple fact that not only are there a lot more kinds of microbes all around, there are simply more microbes in total around us than we can really even wrap our heads around to understand. I mean, even in drinking water that's safe to drink, it is not uncommon for there to be between 10,000 and 1 million microbes in a single milliliter, not even, a cup or an ounce, a milliliter, like a drop. Now, most of them aren't dangerous by any means, and for the small fraction of those that are, this points to the fact of how awesome our immune systems and how hard they work to, every second of every day to protect us. Zooming a little further to water found in fuel systems, bottom water samples. For every drop of water there, you can have up to 1 billion or more cells per milliliter. Now, the reason we mention this is that taking these facts into consideration should make it easier for us to see that there are simply so many microbes in the world surrounding us everywhere we go, every single second of every day. Nobody should ever be surprised when we find them pretty much everywhere, including in fuel systems and storage tanks. Now, since we find microbes in fuel systems and storage tanks all the time, it's fair to ask the question of how they tend to get in there. Now, since we just said they're already in there, what we're really asking is how do they get in there to the extent that they cause problems, I guess is a better way to put it. Now, when you're talking about microbes getting into fuel systems, there are two considerations here, two areas to look at. How do they get into storage tanks, and how do they get into the broader distribution system? Well, for storage tanks, there's lots of ways. Anything that enters a storage tank will have some level of microbial growth associated with it, whether it's the water vapor and the dust, which you can't even see, but it's in the air that's going in and out of a vented tank. There's tons of microbial presence on that. And not to mention if you get water ingress, like a couple gallons of water from a spill bucket that gets an underground tank gets opened, the water rushes in, that water's teeming with microbes. Now, as for the field distribution system, everything upstream of that, you start with the baseline that when the fuel's put together at the refinery, it starts from a sterile state because you've got high distillation temperatures that are used to produce the feedstocks and sterilizes them. But it is only sterile for a short period of time because once it's made, it's going to go into a refinery bulk tank, and it's going to go into a ship or a pipeline or a rail car, which sends it to a fuel depot where it goes into another tank, and before it's sent out to local and regional distribution centers, and it gets aggregated with fuel from, let's say, 100 other refineries. And if any of their tanks have microbial presence in it, now that microbial presence has been carried and aggregated with the 99 fuels from the other 99 places. Not to mention, especially on the refinery level, if you have large fuel storage tanks that have those floating roofs that move up and down as the fuel level drops, those roofs actually scrape microbial presence from the outside that was on the tank shell. It actually scrapes that down into the fuel. So rust and biofilm then accumulates and gets scraped down into the product during each cycle. Now, ultimately, we are most concerned with downstream fuel storage tanks and what the microbes do there. So in these tanks, where do microbes live in these kind of storage tanks and fuel systems? Well, when we talk about microbes in storage tanks, we are accustomed to talking about how microbes like to live at the interface between the fuel and the water. But most commonly, though, that's not their favorite place. Their favorite place, their preferred place, I should say, they grow best within biofilms that are located on solid surfaces. Now, if you look into any storage tank, most storage tanks are going to have microbial presence. A lot of them are going to have biofilm, but that biofilm is not going to be evenly distributed. It's going to be concentrated in certain areas. And where it is concentrated, well, it depends on a number of factors, including what kinds of microbes are in there and what they best like to, what's already in the tank that best feeds what they need to grow. So takeaway there is that biofilms tend to be unevenly distributed in fuel storage tanks. Now, if we were to see some biofilm and we were to zoom down in close, get right down in there, maybe even shrink ourselves in size and get right down in there, would there be anything notable that we would notice about the microbial communities that are present there? Well, if we could zoom down close enough, there are things that we could see about those microbial communities, things that we never would have guessed otherwise. The big thing that we've noticed is that these communities are a lot more complex than we give them credit for. So let's think about how these communities develop. And that's fair for us to consider because it is the communities within the biofilms that cause problems. Individual microbes don't cause problems. It's always the complex communities together that do. So what generally tends to happen is you'll have one free-floating cell, also referred to as a planktonic cell, and it's in liquid. It goes out and it finds a surface to settle on, and it settles there, and then it secretes adhesive substances that help it stick to the surface, and then it starts reproducing. How fast it reproduces depends on what it is, but it's going to reproduce, and it's going to form a microcolony. Then in addition to this, those adhesive substances that it produced, well, those are actually going to cause additional or other planktonic cells to stick to the surface as well, to join it. Then they start reproducing. They're producing their own adhesive substances, and eventually what you get, you give it enough time, and you get the formation of a complex, mature microbial community. Now, how quickly can this happen? This is the surprising thing. All of this can happen from soup to nuts within 24 to 48 hours in a tank. So, going back to biofilm. Contrary to our perception, these slimy masses that we call biofilms, they are not uniform. They're complex, both in internal composition and, of course, in their distribution within a storage tank. In fact, the biofilm that you typically see within a tank can actually have significant variance in its thickness that you would never know unless you had the ability to measure it. Now, what that means is that different parts of one biomass formation, so to speak, can give you vastly different pictures of microbial presence in the tank. So basically, even within one patch of biofilm, you can have, in different areas of that one patch, you can have markedly different kinds of microbes growing and reproducing. So if you were to take a sample from one end and then take a sample to the other and then culture and grow those microbes, you would find many times find a markedly different distribution of microbes. Now, why we're mentioning this is this speaks to the principle that we will touch on multiple times during this webinar, which is where and how you sample controls what you detect and what you see. So within a biofilm, there is a diverse assortment of microbes, a heterogeneous assortment. They exist in symbiosis with each other. There's different kinds of microbes in different locations of that one biofilm. One reason why this happens is because the environment within the biofilm is actually a lot different than the environment outside in the open air. There's greater concentrations of oxygen and sulfur. There's differing levels of acidity, differing levels of carbon dioxide, for example. And if you consider that a biofilm sheet is not uniform all the way through, that means that you could go to one area and it have even different oxygen level and different sulfur. And so that causes a different flora of microbes to grow and thrive there. So ultimately, the important point to take away from that is where you sample can give you different pictures of what you're going to see inside of that tank. Now, naturally, we would expect all these different kinds of microbes may skew towards producing more of one kind of waste product or what we call a metabolite than another kind. But generally speaking, they produce categories of metabolites. They produce weak organic acids, which can many times react with things like aggressive chlorine ions to form strong acids. They form carboxylic acids and acidic alcohols and sulfates and salts, all sorts of things. Now, the one in the middle, biosurfactants, those are important. If you remember from chemistry class, a biosurfactant or a surfactant is a molecule that has a electric charge on one end and no electric charge on the other. Now, the electrical charge is called the polar end, and that's attracted to other polar molecules that have electrical charges, like water, for example. The nonpolar other ends are attracted to nonpolar molecules like fuel. So what happens with these is microbes will produce these, and they will actually utilize these biosurfactants, or what sometimes are called Zwitter ions. They'll actually use them to help disperse water up into the fuel, which makes it easier for the microbes to get at the food molecules in the fuel. Now, speaking of this phenomenon, speaking of emulsifying water and making hazy fuel, because when that happens, it makes the fuel hazy. So speaking of that, now we touched on something key that we need to look at. What effect do microbes have on the fuel itself and on their storage environment? Well, generally speaking, what effect the microbes have on fuel depends on the fuel itself. Now, let's take gasoline, for example. Now, for years, the industry thought that microbes did not like to grow in gasoline because the industry thought that the lighter or the lower end hydrocarbons that were in the gasoline mix, we're talking about the 4 carbons, the 5, 6, and the 7 carbons, C4 through C7. They thought that those were actually toxic to microbes. Now they know that this was not even close to being right. They know now that there are plenty of microbes out there that love to actually consume these. They chew them up. They eat them for fuel. Now, they also these and other kinds of microbes also love to consume the oxygenate in the gasoline. Now, what effect does that have on the fuel, on the gasoline fuel? Well, and also on diesel, but this is more of a gasoline problem. If they consume a portion of the low molecular weight molecules, then what you now have is a change in the molecular distribution of the fuel. You also have a change in the percentage of aromatics because these lighter molecular weight components happen to be aliphatics. Microbes like to use aliphatics for fuel. If they consume a certain portion of the aliphatics, you now have a greater percentage of aromatics proportionately in the fuel blend. Now, the effect that this has on the fuel itself, well, it can change the vapor pressure, and it can certainly change the viscosity. Point is, it can substantively affect the physical properties of the fuel. Now, what about diesel fuel? Uncontrolled microbial growth in diesel fuel also can have specific effects on the physical properties of that fuel, which we will get into more detail a little bit later. Now, how do microbes use the water phase that we're talking about? What effect do they have on the water phase? Maybe that's a better way of putting it. Well, we all know that microbes need some kind of free water to grow. Now, that doesn't mean that if there's no water around they can't live, they probably go dormant. But in order to grow and multiply, they need some free water. But the thing is that they don't need very much at all. If you just have like a 2 millimeter water phase, that is like, proportionately speaking, that water depth, it's like a mountain, like a Mount Kilimanjaro to a microbe. If you're comparing the sizes together, it is exactly like if a human being stood at the base of Mount Kilimanjaro and looked up at the top. That is, proportionately speaking, similar to the size difference between a microbe and just a 2 millimeter water phase. So what happens when a water phase develops under the fuel? Well, what happens is first, because the water is polar, then certain polar molecules that are in the fuel will want to gravitate towards the polar phase. So they will start moving from the fuel into the water. Microbes are there, say thank you very much. They use those for food. The microbes then also produce these biosurfactants. These biosurfactant molecules then help water to actually move up and emulsify into the fuel. And what you get is an invert emulsion, a layer of invert emulsion. Now, if we were to stop there, we'd say, okay, we got hazy fuel, that's not good, but okay, whatever. But the microbes then can move up into the invert emulsion, so they're now up into the fuel phase, or more into the fuel phase than they were before. They're closer to the surrounding fuel molecules. They can use them for food, and there you go. So that is one way that microbes use this water phase to their advantage. And no talk about microbes would be complete without examining the effects they have on the tanks, too. And when we start talking about storage tanks, we start talking about terms like fouling and corrosion. Now, when we talk about storage tank damage, we are used to it being primarily defined as corrosion. But we know that corrosion has to do with reactions with metal. And there's plenty of damage going on with non-metallic parts of the tank. So let's talk about non-corrosive damage. Non-corrosive damage and issues are characterized by things like fouling. Fouling would happen when microbial slime would accumulate on certain fuel system surfaces, whether it's in a filter or if it gets into or on a tank float, it can actually change the float's specific gravity, and that causes the float system to give an inaccurate fuel volume reading. Microbes can also damage tank linings. If you get a pinhole, one little pinhole compromising in the tank lining, it's plenty big enough for fungus and moisture to go into that pinhole. Now, the moisture goes into it, and then when the temperature changes, temperature goes up, temperature goes down, the moisture will expand and contract and expand and contract. And over time, it will start to compromise or delaminate the lining, cause it to become debonded from the tank shell. It's going to blister. It's going to peel away. Of course, at the same time, the microbes, especially the fungi, are also going down into that pinhole along with the moisture, and they're growing and multiplying, and so they work together with the moisture to damage tank linings. Not to mention, there's the issue of the effect that fungus has on fiberglass. See, fungal molecules or organisms, rather, they don't even need a pinhole. They can just colonize the surface of fiberglass. They send out their little hyphae, which penetrate into the fiberglass. They start consuming the resin. They start deplasticizing the fiberglass, and then so you get a major compromising of fiberglass tank surfaces. So there are plenty of ways that non-corrosive damage can happen in storage tanks. But we do have to talk about corrosion itself. Now, before here, we have done trainings on microbe-induced corrosion, what they call MIC, and the effects that MIC has on metal tank components. And there are multiple mechanisms that can happen with that. The most prevalent mechanism for corrosion in the wetted area, not in the vapor space, but in the wetted area, involves formations of these galvanic couples. When you get a galvanic couple, you get movement of electrons from one side to the other. That helps cause corrosion issues. Now, if you get biofilm layers, when you have a biofilm layer, then you get the creation of an electropotential gradient between the covered surface, the surface underneath this piece of biomass, and the uncovered surface that's not covered by the biomass. So you get a difference in electrical potential that leads to galvanic corrosion, which leads to pinhole leaks and damage on steel surfaces. Oxygen content can also cause corrosion because, contrary to what most people think, fuel actually can hold a greater amount of dissolved oxygen than water can. So what happens is you get an osmotic potential situation. Osmotic potential refers to the phenomenon where molecules will want to move from an area of high concentration to an area of low concentration. Well, if you have a water layer and a fuel layer, but you also have some biomass that's nearby, then what you're going to get is you're going to get oxygen diffusing from the fuel into the water. But if it happens around biomass, then some of that oxygen can get trapped by the biomass, and you get a pocket of high oxygen concentration, and that leads to corrosion. Same thing can happen if there's carbon dioxide, that can form carbonic acid. If there's H2S, if you've got sulfate-reducing bacteria, that can help produce sulfuric acid. If you look at the tank water bottoms and you look at the sodium levels, if those are high enough, then those can react and form chemical soaps. And what the chemical soaps do is those go out, and those actually deactivate the corrosion inhibitors in the fuel itself. So multiple ways for corrosion, or multiple mechanisms for corrosion to happen in these tanks. Of course, no discussion of microbial-induced corrosion would be complete without talking about the acid-producing microbes, the acid-producing bacteria. Acid-producing bacteria, they produce things like short-chain fatty acids. They also produce low molecular weight acids, the light acids like acetic acid and formic acid and lactic acid. Now, not only do these lightweight acids volatilize and go up into the vapor space and cause vapor space corrosion, but they also themselves provide nutrients for other kinds of microbes like sulfate-reducing bacteria. There are still other bacterial species that produce themselves aggressive inorganic acids like sulfuric, which, as we know, anytime you have sulfuric, you're going to have corrosion. Or those can contribute to the production of these localized high concentrations of carbon dioxide, which, if there's any water present, they will dissolve in the water, and that will produce carbonic acid, which is highly corrosive to steel, of course. Now, how quickly does this happen? The literature value is for diesel tanks between 0.3 and 0.5 millimeters per year is common. Gasoline's about a third that. And part of the nature of tank corrosion damage and fouling and microbial-induced corrosion, part of the nature of this is that you're rarely going to find it unless you're actually looking for it. And that means you rarely find it until it has developed to the point where the problems are serious and obvious. And this point has important implications for stored fuel managers. Once the signs of corrosion show up, the damage is already well on its way to having been done. So the goal here is to head off the problem before it develops, before it becomes expensive, which is why we have to consider the issue of condition monitoring here. Now, the object of condition monitoring is to develop an accurate picture of the condition of your storage tank and the contents therein. That means taking samples of the contents and then analyzing those samples to see if they tell us anything useful about the situation. Now, there are many ways to do these things, and unfortunately, there's many ways to do them wrong. And there's many considerations that need to be accounted for if we're going to do them right. Now, microbes in fuel and storage tanks, they often go undetected because either you got incorrect sampling, or you got incorrect testing, or you may have a combination of the two. Now, of those two, sampling is probably the greater problem. So, accurately detecting microbial contamination starts with good sampling. Now, it is human nature that people always assume that their test is telling them what they really want to know. And they assume that because their test was done on a sample they drew, they assume that their sampling is an accurate representation of the actual condition of their tank. But remember what we said. Where and how you sample really does control what you detect. Let's use an example. Most commonly at a fueling station, fuel samples are going to be collected from dispensers because, well, it's fast and it's easy, it's convenient, and it gives a nice, good fuel sample that is perfect for fuel quality sampling, fuel quality testing, but not so good for microbial testing. So if you're relying on a dispenser sample to test for microbes, you're not going to have a representative sample at all, and you're not going to find out what you think you need to know. Now, even issues like with tank design, an issue as simple and yet as out of our control as tank design and tank settling, those can have major impacts on our ability to detect and remove water, which essentially means they are integrally tied to microbial growth issues. See, it's not uncommon to take a fuel tank and check for water and have zero detected free water at the fill end, and yet down at the turbine end you've got like 50 gallons of water sitting there that you didn't really know about. Now, when you're putting underground storage tanks, when you're putting them in, the regulations dictate that the tank has to be trimmed by the fill end. That means it either has to be level or slightly lower on the fill end. But yet, because of their weight, tanks almost always settle into their backfill, and many times they settle in certain ways that influence where the water is going to collect at. Now, this can also be something even as simple as over time, because of the simple weight of thousands of gallons of fuel, the bottom plates of the tank actually get deformed, and they change shape because of that tremendous amount of hydrostatic pressure. So they change shape relative to the shape of the overall tank, and it creates pockets, spaces where water will pool and collect. Now, how do you know this? How do you know if those have deformed? You really don't. So these are really examples of how much variance that there can be between what's appearing in our sample and what's actually going on in reality in the storage tank. Now, if it's true for something as simple as water in the tank, it's just as easily true for microbes as well. And without a good sample, it is impossible to know if your microbial test results reflect the reality of the situation in your storage tank. But who defines or who tells us how to get a good sample? Well, the gold standards for sampling practices are the ASTM standards. ASTM 4057 is the standard practice for manual sampling of petroleum and petroleum products. This covers how to best collect fuel samples if you want to do specification testing, like fuel quality characteristics. The problem, though, is that it doesn't cover the special issues that have to be taken into consideration if you want to test for microbes. So that's why they made the 7464. 7464 basically fills in the gaps and gives additional guidelines on how best to do sampling that's intended to be used to do microbiological determination. Now, remember earlier we said that if they had their way, microbes would most like to live on solid surfaces within some kind of biofilm. So, if your goal is to sample or is to get a sample that is most like what's actually in your tank, then you would want to do surface sampling if you could. Unfortunately, it is rarely practical to get a surface sample, which means we have to go to our next best alternative, which are bottom samples, specifically three-phase bottom samples. In the sample, you should have some bottom water, you should have some rag layer, and then you should have some near bottom fuel, which is fuel that is between 10 and 16 inches above the water phase. And this means to get this kind of ideal sample, you need to check for water beforehand so you know how high your water phase is, so you know how high 10 to 16 inches above that is. Other points that are pointed out in these ASTM guidelines, you want to make sure you sterilize your sampling tools and equipment. Basically, any surface that you anticipate is going to come, any surface that you anticipate your fuel sample is going to come into contact with before it ends up in the sample container. Best practices for collecting these samples is, first of all, put samples in unused bottles, and then before you collect the next sample, you need to rinse your sampler and all those other transfer tools, rinse them in alcohol. That's at least 70% alcohol by content. You do that in between each sample collected. And if you're using a funnel to transfer fluids, you especially want to make sure that it is sterilized. So, good sampling techniques are essential to getting as accurate a picture of the realities of a storage tank as possible. Now, there are other considerations too, beyond just best practice techniques for pulling samples. Condition monitoring also means getting enough data so that you can build a fingerprint view of what your individual tank looks like. Now, when we're interpreting microbial data here, we use general benchmarks to give them meaning. Like, for example, a benchmark, general benchmark might be if you're over 10,000 microbial units per milliliter, that typically indicates a problem. Okay. But when you get down to it, every tank environment is different. And 10,000 MEs in one kind of tank might be a problem, but in another kind of tank, it might not be a problem. Every tank in every place has a fingerprint. And the only way to get the right fingerprint for your specific tank is to do trending. And trending requires taking multiple samples over a period of time. That is a key part of what goes into condition monitoring. So you start by getting a baseline. The baseline is your benchmark. Ideally, you want to collect the baseline just after the system is cleaned, if you can. Then, after you establish the baseline, you want to sample. Best practice is to sample at least 3 times during the control interval. Okay, control interval, what are we talking about there? Well, control intervals are important because as you get more data on your tank, what you start to see is the points in time where the microbe levels rise and fall. And we say rise and fall because this is not a completely linear phenomenon here. Sometimes they'll go up, sometimes they'll go a little bit down. In general, they tend to have a trend where they rise over time, but it's not completely linear. So, over time, you see your microbial levels rise steadily, and eventually you see the point in time at which they exceed the threshold for defining, hey, we've got a problem, we've got to do something about this. And that point in time is your control interval. It's basically how long does it take for microbes to rise to problematic levels in a given tank. Now, we've been trying to stress that every tank is different. However, if you want to generalize this situation, then a common control interval for bulk storage fuel tanks is about 6 to 12 months. So what that means is that in order to get enough data to have good trending, you're probably going to want to sample and test at least every other month. So you need to get enough data to figure out what your true control interval should be. Now, quick note on the subject of chain of custody. In any condition monitoring situation, it is always best practice to have chain of custody documentation whenever samples are being collected in order to be tested at a later time and place. Now, a typical chain of custody form is going to give you enough information to document who had the sample at what point in time, what is to be done with the sample, and who did that work. So, chain of custody, documentation for samples, always best practice. So having covered the important elements of condition monitoring, we switch our attention back to the samples themselves. We've already talked about how to pull a good sample, but a good sample can become a bad sample if it is not properly handled. Microbes are living organisms. When you sample them and you take them out of their normal environment, they can react in ways that weaken the relationship between your test results and the state of the system that they came in. When you pull a sample, over time, some microbes in that sample might die off, in which case you get a negative test result, or I should say a false negative test result. Some of the microbes might like your sample better, though, and they might multiply so that you end up with more microbes than you actually had at the beginning. If you're lucky, though, you won't have too much of either, and your microbial population won't effectively change. In other words, the longer the period of time passes between sample collection and testing, the weaker the relationship that you're going to have to acknowledge. So, how much time are we talking about here? Well, the ideal situation, ideally, is if you can pull a sample and test it within 4 hours, that's ideal. However, in the real world, more than 80% of samples that we get to test are older than 24 hours. So we always have to take this into account. We always have to have the possibility of margin of error and take that into account in our interpretation of what we find after we test our samples. And that is a good segue because since we've covered the important considerations for condition monitoring, now we can turn our attention to what follows naturally from that, which is condition testing. So up to this point, we talked about microbes, right? We talked about how they grow. We talked about what they do and how they do it. We talked about how to monitor their presence and their development in tanks and systems. And we talked about the importance of good sampling techniques in enabling us to do that. So it's now appropriate to transition from monitoring to testing. Now that we have what we hope are good and accurate samples, our goal now is to get the samples to tell us the right story, which means we have to ask the right questions. That is kind of a fancy way of saying that we need to have the right testing approach to figure out what the samples tell us about where they came from. Now, from a fuel microbiology standpoint, those questions are going to be along the lines of, number one, do I have microbial contamination in my storage tank? Yeah. Number two, how much microbial presence do I have? And number three, is the presence that I do have, is it contributing to existing problems, or how likely is it to do so in the future, basically? So these are the kind of considerations that fuel microbiologists hope to clarify as they move into this field of condition testing. Now, for condition testing, there are 4 broad categories that will give us the information that we need. The first one is gross observation. Second one is physical testing. Third one is chemical testing. And the fourth one, last but not by any means least, is microbiological testing. So let's delve into these categories and talk about what kinds of tests go into them and what roles or what values they may have towards helping us answer the right questions that we're trying to ask. So in condition testing, we typically start at the simple end. And the simple end is gross observation. Now, there are observations about fuel that we can make with our senses without having to rely on laboratory procedures. So you look at the fuel. Does it look different? Are there characteristic odors that are different from how fuel's normally supposed to smell? If you have a filter and it's got a coating on it, what does that coating feel like? Is it a dry particulate or is it a slimy kind of coating? More specifically for visual inspection, looking at the fuel, there is actually a guide that we can use that helps to standardize the results. That is ASTM D4176, called the Visual Inspection for Water and Particulates. And it has two procedures that can be done. The first one is commonly referred to as like a vortex or swirl test. They have a sample, they swirl it for about 30 seconds, so you create a vortex, and then you hold it up to the light and you observe what you see. Now you're going to see air bubbles that are going to rise to the sample surface, but if there's any free water and if there's any particulate, those will actually sink to the bottom. And if the technician sees any of those, then that's going to be a failing test for that. Now, that one's good for like dispenser samples, not so good for bottom samples that already have known particulate and water in them. But procedure 2, the clarity test, that one is useful for all kinds of fuel samples. You take a specified fuel sample in a clear container, and you hold it up to a bar or a line chart such as what you can see on your screen. Starting from top left through bottom right, you can see the different visual clarity scores. You give it a score from 1 to 6 based on the number of lines you see. So 1 on the top left, that's the most clear. Number 6 on the bottom right is the least clear. So if you have a fuel sample and it looks like number 4 or 5 or 6, that's a typical good indication that there is something else going on with that fuel. So changes in the normal physical or sensory properties of the fuel are a hint that something's going on. We also know that microbial contamination will change the physical and chemical properties or specifications of the fuel in certain distinct ways. So our next step for condition testing beyond gross observation might be to look at some of the tests that we could use to document and measure the properties of the fuel, as well as trying to look at them to predict whether the fuel will change in the future. Now, there's lots of ones we could run, but the most important tests out of these to look for are the stability tests: accelerated oxidative, accelerated thermal, and petrooxy, which is petrooxy is like oxidative stability, but it's kind of optimized for ultra-low sulfur diesel. Now, one thing to remember with stability tests, they are predictive tests. They predict via some kind of score. They try to predict whether the fuel sample's properties will change in the future. And we know that if there's significant microbial presence in the fuel, it will produce unstable precursor molecules, which makes that fuel more likely to change condition in a shorter period of time. Thus, stability tests like these can give us an analytical hint about whether there might possibly be microbial contamination going on. Now, there are other physical tests that we might be able to use to assess the state of the fuel, and not just simply assess it in a vacuum, but assess it with respect to judging whether we can infer microbial contamination. Some of those, a few of those are on the screen. Filter blocking tendencies. This is basically a measurement of the particulate load in a fuel, which, if you have high microbial contamination, you're going to have higher particulate load. Kinematic viscosity. Remember, we talked about the fact that microbes can change viscosity because they consume lower end hydrocarbons. So if this test is off, that can be a hint that something's going on. Water separation properties. We talked about microbes producing biosurfactants. Well, if the fuel has a poor score of water separation properties, that can hint at possibly there being a microbial contamination where this has happened. And then there's other tests you can look at, like Karl Fischer water content, peroxide number, fuel corrosivity, total acid number, and of course, no assessment of physical or chemical characteristics would be complete without looking at some tests specific to the associated bottom water, specifically pH and total dissolved solids. Now, There are other physical tests that we could do, but these are ones that are the most useful. And that means for condition testing, we have now covered gross observations that can hint at microbial contamination. And we've looked at physical and chemical tests that can give us a more measured confirmation of the properties of the fuel, which can then hint at the possible presence of something that has changed the fuel's properties. Now, both gross observation and physical and chemical testing have this in common. They can hint at the possibility of microbial contamination. But the last category of tests that we would undertake in a systematic condition testing program are the tests that are most specific to measuring or detecting microbial contamination itself. And those are the microbiological tests. This is, now we're getting into the good stuff here. There are three types or three classes of microbe detection tests that are most widely in use out in the field. Now, this doesn't count highly specialized genomic tests that cost thousands of dollars and are only done in a specialized genetics lab. Now, the differences between these three options for microbiological testing, recognizing those differences should illustrate to us an important point, a point about how different kinds of tests only give us differing fractions of the picture. Some tests are better at this than others, but really we have to recognize that any test is only going to give us a sampling of the true situation. What we want to do is we want to select a test that gives us the best fraction of that. So first one, culture tests. Culture tests, they're the oldest microbe tests. They date back to the mid-19th century. They have an advantage in that they're pretty simple to administer, which is probably a big reason why they're so widely used. But they do have limitations that by nature affect their utility for what we're really trying to do, which is, of course, get an accurate picture of what's going on. Some of these limitations, remember the fraction thing. Culture tests only detect a very small fraction of what we would call the microbiome, the entire microbiological picture of a given environment. And this is because culture tests depend on the ability of cells, microbial cells, to grow under the culture conditions. And unfortunately, those culture conditions many times can be significantly different than the environment that we took them from. So 0.1% accuracy. Number two, contamination is a big problem. Again, culture tests rely on us being able to grow a representative sample of the microbiome. But it is surprisingly easy to get outside organisms in the sample. And then they grow alongside the ones that are supposed to be there. They produce their own colonies. But in the end, you don't have any idea which ones came from the outside and which ones were yours to start with. And then third one, culture tests, you have to give them time to get results, typically between 3 and 5 days. Now, that is a severe limitation to their usefulness out in the field. If you wait 3 days for tests to incubate, how do you know if what you're seeing really represents the microbiome if you got outside microbes in there and you gave them 3 to 5 days to grow and multiply? Now, for those looking at commercially available culture tests, there are some options, some of which are better than others. One that we don't have on here is the paddle slides, the dip slides, because really I'm trying to think of a nice way to put this, no self-respecting condition monitoring or fuel maintenance program is going to use paddle tests. They're just too problematic. Now, Microbe Monitor 2 has an ASTM number associated with it, 7978. This is what the person at the top is handling, the person with the blue gloves. It's a container that has this thixotrophic culture gel. And so you inoculate the bottle with your fuel, you let it grow, you count the number of colonies, and you compare that to a chart in order to give a general range of number of microbial presence in your fuel. There's also LiquiCult. LiquiCult is also known as non-selective broth. It contains a dye that turns red as the microbes grow and produce acid, and so it turns red. And so at a given point, you leave them for X amount of time, and then you have to check them. And then depending on what the number is, not the number, the color, they give you the ability to interpret what that corresponds to with respect to the amount of microbial presence in the sample. Then that last one, a BART slate, I guess I would call it, are kind of like liquid cult, except they're multiple bottles. They're all packaged together, and they contain different kinds of growth medium that grow different kinds of molecules. And so that actually has some usefulness because it can actually help you to see what kinds of microbes, whether it's a fungi, or whether it's a sulfate-reducing bacteria or a slime-forming bacteria. It helps you to see a little bit more specifically what kind of microbes you have in your sample. So those are the culture tests. Now the second category of microbe tests are what they would call the lateral flow devices, or LFDs. Now this is a fancy name for what they would call immunoassay tests. And these are tests that measure the amount of small molecules in a solution by using some kind of antibody substance. Now, the dominant LFD microbe test on the market is the FuelStat Resonate test by Canidia Bioscience. It is associated with ASTM 8070. When each test looks like what you see on your screen, there's 3 sets of modules, paddles, I guess what you would call them. And so you prepare your sample, you put drops in on each of the 6, and you wait to see how many lines show up. And then depending on the number of lines that show up, that should tell you if you have low or medium or high levels of different kinds of bacteria and fungi, and then last one of a specific one. I want to say that it's I can't remember which one it is. It's not Pseudomonas, but it's Helicobacter H. resinae, basically. It's a specific kind that they have determined or they think is common in fuel systems. Now, good thing about it is it's about 60% accurate, which means it's 600 times more accurate than the culture tests. It also works reasonably fast, which means you don't have to wait 5 days. You get a result in about 5 minutes. The downside, though, is that it can't be calibrated, which means if you don't get a result, do you know if it's because you really don't have microbes, or is that because your test went bad? You improperly stored it or something. It does not give you a specific number of microbes. It just gives you a range, and unfortunately, having executed a number of these tests, it can be confusing to properly interpret. And then lastly, it's expensive. It's between $100 and $150 a shot, and especially if you can't calibrate it, that's an awful lot of chunk of change to go down the drain if you do it wrong or if the test has gone bad. And the problem is you wouldn't know if it went bad. So, last ones, ATP tests. This is the one that we are most concerned with, ATP testing. This corresponds with ASTM D7687, measurement of cellular ATP in fuel and water with sample concentration by filtration. Now, ATP testing itself was developed in the 1950s. So it's been around for a little while, and it is based on the principle that all living cells use ATP molecules to transport energy. We also know that all microbial cells tend to have a relatively constant amount of ATP on them. So if you could isolate all the ATP from the cells, extract it, and then measure it, you could calculate the number of living microbe cells in your sample. Now, the process itself is a multi-step process, and the very basic explanation is on the screen. Basically, what you're going to do is you're going to filter out all the microbes from the sample. You're going to wash it, wash out the impurities. You're going to dry it, You're going to extract microbial ATP using a special solvent. That gets diluted, and then you put a measured amount of that and an amount of a certain kind of enzyme that produces light when it's exposed to UV light. It glows like a firefly, basically. So then you take that little test tube, you put it in a luminometer, which exposes it to the light, measures the luminescence, so to speak, and from that you get fairly accurate results on the microbial content in that sample. Now, just hearing about what it's supposed to do, there are some obvious and huge advantages to this. Like the LFDs, you can do it, it's fast. From start to finish, you can do a test in less than 5 minutes. In fact, the actual processing of it by the luminometer is like less than 10 seconds. It's the most accurate test you can get. It detects about 70% of the microbes. And the big time advantage is that it's quantitative. That means it gives you a number. It just doesn't give you a range. And this is really, really important if you're doing something like trending, because you can use ATP testing to trend the numbers over time. And you can't do that with culture tests, and it's really hard. Really functionally, it's impossible to do that with any other kind of test other than this kind of test. So one of the takeaways from all this is that we have to keep a proper perspective on the accuracy and the meaning of the microbial test results that we get. This is because despite what we may want to believe, a positive or a negative test may not tell us the entirety of what we think it's telling us. For example, you have a culture dip slide, culture test, comes back negative. Do you know for sure that this means there's no microbes in the tank? The correct answer to that should be not necessarily, because we have to acknowledge the possibility that we could have a false negative. Now, you get a lot fewer false negatives with ATP testing than you do with like a culture test. But even acknowledging that, we do have to acknowledge the fact that with an ATP test, you're actually getting a number of all microbes together. Now, when we talk about this, we say not all reds and greens are created equal. Now, what we mean by that is when we do ATP testing, we get a total number of microbes. You get a number, right? And then that number is compared to recommended ranges. And the recommended ranges say if it's below 10, you're green. That means it's not a problem. It's a manageable level. If it's between 10 and 100, you're yellow. That means it's starting to get towards the danger zone, but you're not in a severe contamination point yet. And then if it's over 100, then you're red. You've got enough microbes in there that you've got a severe contamination. You need to do something about that. But we have to remember, though, that this is total microbe count. If you've got 100, that doesn't mean it's 100 problematic. It's just total microbes. Now, so we have to acknowledge that possibility, but we don't really have a way to find out anything more specific than that. So that basically means we have to acknowledge that that's true, but that doesn't entail that there's anything else specific that we can or should do about that. So that is a brief consideration of the testing options that we have available. Now, our goal has been to detect and measure if we have microbial content. Then based on the results, we can transition from monitoring and testing to contamination control, which is controlling and getting rid of the problem that we think we have. Now, the three most common microbial contamination control actions are dewatering and tank cleaning and chemical treatment. Now, we have discussed before how microbes don't need very much water to grow. Now, let's say you have a bulk fuel tank and you remove water. Now, maybe you're able to get it all. You're certainly trying to get it all. But Practically speaking, you're probably not going to get that last 1/8 to 1/4 inch of water. Maybe you get all but like 2 gallons of water in there. And most people think that that's pretty good. Now remember, we said that each milliliter of tank bottom water can contain up to 1 billion microbes. So if you have 2 gallons of water, that's enough water left to support 8.5 trillion microbes. So it is really important to get as much bottom water out as possible. But it is also, we have to have the expectation that we're not going to get it all. And the simple fact that bottom water has so much concentrated microbial presence, that means we have to do more than just remove the water if we hope to solve the problem. We have to do stuff like tank cleaning. Now, there's different kinds of tank cleaning. Man entry is the gold standard, but it's expensive and it involves high insurance rates, and it's comprehensive, but it's expensive. And there's not a lot of companies that have the ability or the desire to really do that. So what most companies default to is external tank cleaning, where they'll have a fuel polisher or a tank cleaner come out and use stingers and suctions to suction out as much of the water as possible and as much of the biofilm as possible. Unfortunately, we know that because of the way biofilm is distributed, you're probably not going to get it all, but you try to get as much of it out as possible. Now, and this is really important because remember, we said microbes love to live in the biofilm, so this is, it's important to get as much of that out as possible. If the greater amount of biofilm that you leave in there, the higher the chance is that your next thing you're going to do, your chemical treatments, it increases the chances that it may not be fully effective. So you want to have a good thorough tank cleaning and a good thorough dewatering. And then the last thing is chemical treatment. You've gotten as much water out as you think you can. You've removed as much biofilm formation as possible, but you know that you didn't quite get all of it. So now you treat with a biocide. Now, with Bellfield and Tank Services, we have 2 chemistries that we use for biocide treatment. Bellicide is the thiocyanate. Bellicide ISO is isothiazolone. Both of them are what they call broad spectrum. It means they're pretty good at killing as many different kinds of microbes as possible. Once you have the effective chemistries, you have to make sure you use them properly. Biocides need time to work. You can't just dump a biocide in and then, your problem solved 5 minutes later. It doesn't work that way. You apply biocide, you get it mixed in, you leave it for a period of time, which is what professionals call the soak time, and optimal soak time is 24 to 48 hours. For small tanks, 12 hours can be optimal. And of course, if you have a really, really heavily infected system, then you would plan to see plugged filters until the killed biofilm has been flushed out from the system. Alternatively, if a fuel polisher is using this, then they would apply the biocide, they'd give it the soak time, and then they would filter everything, and that would take care of that problem. Now, in all of this, it is essential to keep in mind that not all tanks are the same. See, in the rush for the industry to simplify solutions, sometimes that's a disservice that's done to the customer. This implication that one size fits all, in the sense that no matter how bad the extent of your tank infection is, one shot of biocide is going to cure everything every time. But there are cases where you may need to do 2 treatments of biocide. That happens. And people need to recognize that there is the possibility that that will happen. Not all tanks are the same. Not all contamination levels are the same. And even though we just said that our biocides are broad spectrum, we still have to acknowledge that not all kinds of microbes respond exactly the same to every kind of biocide chemistry. So it is important to be able to do things like condition monitoring. See, condition monitoring is really important because without having a tool like ATP testing, where you can actually measure your before and after microbe levels. You're really only guessing at whether you solved the problem. Now, whether you're going to need a multiple biocide application or not, many times hinges on the level of contamination. There are some tanks that will require 2. Rarely will they require 3. So most tanks, some tanks may require 2 applications, but how would you know? How would you know? Only if you're doing ATP testing where you're measuring the microbial levels can you determine if the microbial levels have actually been reduced to your desired level. Then, this is also where pairing that biocide with a biomass dispersant like tank treatment SCS, that can also be useful. It disperses biomass, it breaks it up, it increases the efficacy of the biocide because it enables more of the microbes to be exposed to the biocide, which is what you need. Okay, so we have covered an awful lot over the course of these trainings. Hopefully, besides some useful tidbits about field microbiology and the important aspects of it that inform what we do with Bell Fuel and Tank Services, hopefully you will also come away with a sense that the general issue of microbes in fuel is a lot more complicated than it may seem at first. Microbial contamination control for tanks and fuel systems is a never-ending process. It is not a fix once and you're done forever process. It requires us to pay attention, both in preventing it and fixing it when we find it. In other words, one size does not fit all when it comes to managing stored fuel. And our goal with Bell Fuel and Tank Services is to make the process of condition monitoring and condition testing and contamination control, make it as easy and cost-effective for our customers as possible. So if you have any other questions, visit bellperformance.com and click on the Bell Fuel and Tank Services link for more information on everything that we have discussed here. I am Eric Bjornstad, Technical Information Director with Bell Performance. Thank you for joining me for this training on fuel microbiology made easy, and we'll see you next time. Bye.